Hello friends, welcome to Unlocked Science by Mishtab. No cramming, just understanding. Still in the world of cytology, but before, did you know that the very first life on Earth was the tiny cells with no nucleus, yet powerful enough to shape our world and make all life possible? They survive in hot springs, frozen oceans, and even inside plants and animals. These are the oldest and most successful life forms ever, the so-called prokaryotic cells. Therefore, by end of this video, you'll be able to define prokaryotic cells and examples of prokaryotes, their key features and structure, mode of life of prokaryotes, importance of prokaryotes in the environment. So get ready. By end of this video, you will truly appreciate these tiny but powerful living things. Let's dive in. Prokaryotic cells are cells with no distinct nucleus, and their DNA material is not enclosed in the nuclear membrane. Prokaryotic comes from two words. Pro means before, and karyo means nucleus. So prokaryotic literally means before nucleus, and this is equivalent to the lack of distinct nucleus in our definition. Their DNA material is concentrated in a region called nucleoid, but there is no membrane that separates this region from the rest of the cell, and therefore it is spread in the cytoplasm. Organisms containing prokaryotic cells are called prokaryotes, and the prime examples are bacteria and archaea. Archaea live in extreme environments like hot springs, salty lakes, and frozen oceans. Having seen common examples of prokaryotes, let us now explore the complete structure that make up a typical prokaryotic cell and enable it to survive and function efficiently. Parts of a prokaryotic cell. The outermost part of a prokaryotic cell is the capsule, which protects the cell against mechanical damage. Given the harsh environment where these prokaryotes thrive in, this capsule plays a big role by protecting them against mechanical damage. Just beneath the capsule is the cell wall, which protects the inner parts of the cell from mechanical damage and regulates the entry and exit of materials in and out of the cell. The cell membrane also lies beneath the cell wall, and this controls entry and exit of materials in and out of the cell. The cell membrane encloses a gel-like fluid called cytoplasm, which is a medium where all other granules are suspended and most of the chemical reactions take place in this cytoplasm. Then there is a nucleoid, which contains genetic information, and this genetic information controls all the activities of a cell. Prokaryotic cells have ribosomes for making proteins, mesosome, which is a site of respiration, and may also be involved in cell division and DNA uptake. Plasmid, which contain DNA for special traits like survival in extreme environments. Though this plasmid is not found in all prokaryotes, it is found in some prokaryotes which thrive in extreme environments. We have the pili for sensitivity and attachment to the surface, and it is also involved in the transfer of DNA during conjugation. Some prokaryotes that make their own food have chloroplasts, which traps sunlight and aid in the process of photosynthesis. We have the flagellum, used for movement or locomotion, and it's not found in all prokaryotes. Some prokaryotes do not locomote and therefore they lack the flagellum. That's the structure of prokaryotic cell: the capsule for protection against mechanical damage, cell wall for protection against mechanical damage of the inner parts and regulational movement of materials in and out of the cell, cell membrane for regulation of movement of materials in and out of the cell, nucleoid containing genetic information, mesosome for respiration, ribosomes make proteins, flagellum for locomotion, cytoplasm, medium in which all other granules are suspended, and most of the chemical reactions take place there, pili for sensitivity and attachment, Plasmids contain extra DNA for special traits like survival in extreme environments. With clear understanding of prokaryotic cell components, we can now examine how these structures support their mode of life, including nutrition, reproduction, and environmental adaptation. Mode of life of prokaryotes. Prokaryotes are the most ancient and diverse forms of life on Earth. Despite being structurally single-celled organisms, they exhibit a vast range of metabolic strategies, reproductive methods, and adaptation that allow them to thrive in every environment. Nutritional strategies. Prokaryotic metabolism is far more varied than that of eukaryotes, and they can obtain energy and carbon in numerous ways. Based on their nutritional needs, they are classified into four groups. We have photoautotrophs. These capture light to synthesize their own food from carbon dioxide. A prime example is cyanobacteria, responsible for oxygenating the early Earth's atmosphere, since they were the first forms of life on Earth. Photoheterotrophs use light for energy but acquire carbon from organic compounds. Chemoautotrophs obtain energy by oxidizing inorganic compounds such as hydrogen sulfide, ammonia, and use carbon dioxide as their carbon source. Many extremophiles, including those at the hydrothermal vents, are chemoautotrophs. Chemoautotrophs acquire both energy and carbon from organic compounds. This is the most common nutritional mode and is shared with animals. Many chemoautotrophs act as decomposers, recycling essential nutrients like carbon and nitrogen. Oxygen requirements. Prokaryotes also have diverse needs regarding oxygen for respiration. They are divided into three groups. 1. Obligate aerobes which survive only in the presence of oxygen and they carry out aerobic respiration. Obligate anaerobes which survive only in absence of oxygen and they carry out anaerobic respiration. Facultative anaerobes use both anaerobic and aerobic respiration and therefore they survive in both presence and absence of oxygen. Many pathogens, including the E. coli, are facultative anaerobes. Reproductive methods. Prokaryotes reproduce asexually, though they have unique methods for genetic variation. Binary fission is the most common form of reproduction, where a single cell grows and then divides into two genetically identical daughter cells. When conditions are favorable, some bacteria reproduce in minutes, allowing for rapid population growth. Horizontal gene transfer. Although asexual, prokaryotes can exchange genetic material through several mechanisms. The first one, transformation. A prokaryote takes up free DNA from the environment, which may contain genes from other dead prokaryotes. This DNA can contain beneficial genes like antibiotic resistance, new metabolic pathways, and was incorporated into the bacterium's genome. The cell can express new traits, increasing variation in the population. Transduction. Viruses that infect bacteria, that's bacteriophage, or archaea, move genetic material from one individual to another. Conjugation. DNA is transferred directly from one prokaryote to another through a protein tube-like structure called the pilus, and the plural form of pilus is pili. The part we looked at, and it is used for sensitivity and attachment on surfaces, and also in transfer of DNA during conjugation. Those are the three methods in which genetic variation is brought about in a population of prokaryotes. Then the last mode of life is habitat adaptation. Prokaryotes inhabit a vast range of environments, adapting to conditions that would be lethal to most other forms. One, we have extremophiles. Many archaea and bacteria are extremophiles, able to thrive in harsh conditions. They are grouped into two. Halophiles, those are salt lavas that live in highly saline environments like the Dead Sea. Then thermophiles, heat lavas. that live in high temperatures, such as those found in hot springs and hydrothermal vents. Biofilms. Most prokaryotes prefer to live in communities called biofilm, where they interact with other microorganisms. In a biofilm, prokaryotes secrete a matrix of polysaccharides, proteins, and nucleic acids, creating a sticky shield for protection. Biofilms are highly resistant to antibiotics and disinfectants, making them hard to eradicate. Symbiotic relationships. Many prokaryotes live in or on other organisms in a symbiotic relationship. For example, human microbiome consists of trillions of prokaryotes that help in digestion, nutrient absorption, and immune function. The other mode of habitat and adaptation is endospores. Some bacteria can form endospores, which are dormant, tough, non-reproductive structures. They can survive harsh conditions like heat, radiation, and hydration for very long periods, allowing bacteria to persist until favorable conditions return. With the mode of life understood, we can now discuss the significance in nature and human life, highlighting the crucial roles they play in ecosystems, industries, and health. Ecologically, prokaryotes are decomposers. They break down dead organisms and wastes, recycling nutrients like nitrogen and carbon into the ecosystem. Nitrogen fixation. Certain bacteria like Rhizobium convert atmospheric nitrogen into forms plants can use, which is essential for plant growth. Photosynthesis. Cyanobacteria produce oxygen during the process of photosynthesis and are primary producers in the ecosystems, so they make their own food. Medical importance. Normal flora. Some bacteria live in humans, like the guts and skin, and help digest food, produce vitamins like vitamin B and K, and prevent harmful bacteria from colonizing. Pathogens. Some prokaryotes can cause diseases, for example, Mycobacterium tuberculosis, which causes TB and is important in medicine and research. Source of antibiotics. Many antibiotics are produced by bacteria. Industrial importance. Food production. Bacteria are used in making yogurt, cheese, and other fermented products. Biotechnology. Genetic engineering uses bacteria to produce insulin, vaccines, enzymes, and other bioproducts. Waste treatment. Certain bacteria help in sewage treatment by breaking down organic matter. Scientifically, model organisms. E. coli is used in laboratories to study genetics, molecular biology, and evolution. Bioremediation. Bacteria can clean up oil spills or toxic wastes by metabolizing pollutants. And that wraps our today's lesson. Prokaryotes may be tiny, but their impact in the environment is huge. Stay curious and exploring. Don't forget to subscribe. Bye bye.